Okay, welcome to Unit 2, Chapter 7. So after completing these lecture slides, you'll be ready to do homework 2. So this chapter is all about the ways microbes grow and what they require for growth. So growing bacteria in the lab, like any microbe, requires specific conditions. Bacteria grow very well on a surface called agar, which is similar to jello but a little bit more stiff. When you make agar, you can provide all essential nutrients that bacteria require for growth, and the bacteria grow on the surface of the agar and feed off of the nutrients inside it. There are multiple types of agar. You can have broth, which would be a liquid media or agar, as shown in part A of this figure. You also have agar slants, which increase the surface area, so bacteria would grow all across the surface of the slant in B and C. And then in D we have a test tube with a very deep layer of agar, and this will be important for growing microbes that need anaerobic or oxygen-free conditions. We also have the agar plate shown in E, which is a petri dish with a very thin layer of agar on it, and you just spread the bacteria on the surface of the agar, and the bacteria will multiply and increase in number and grow in a thick layer on the surface of the agar plate. When you're making agar or any sort of liquid broth, you have to add some specific things to the media in order for the microorganism to grow. And so shown in this table from your textbook are several of the components that are required for microbe growth. So the ones that have a high percentage composition in the agar are called macronutrients. So these are carbon, oxygen, nitrogen, hydrogen, and phosphorus. And then all of the components that have to be present in small amounts are called micronutrients. And in this table you can see that the macro and micronutrients are important for building organic compounds like those listed, and also occasionally you have to add inorganic compounds to be used in other ways by the cell. All microbes have a preferred growth temperature, so you can divide up microbes into categories based on their preferred growth range. So shown in this figure, we have temperature on the x-axis and rate of growth for the microbe on the y. So as we increase the rate of growth of the microbe is growing faster and growing better. And so, for example, looking at the growth curve for the mesophile, if a microbe is a mesophile, it can grow between the temperature range of 10 to 50 degrees centigrade, with optimal growth being right at 37 degrees centigrade. So the peak of the growth curve would be considered the optimum temperature for each group of microorganisms. So because of the growth ranges of different groups of microorganisms, microbes can grow in basically all parts of the earth. Additionally, microbes have a preferred growth pH. So some microbes prefer a neutral pH, where others would prefer slightly acidic or slightly basic. So in this image, we have a neutral pH petri dish on the left, so we're at pH 7.3. And this is where a lot of bacteria growth will occur. If we lower the pH of the agar in the petri dish to be slightly basic at pH 5.6, we'll see preferred growth of fungal colonies instead of bacterial ones. And any organism that is preferentially growing in an acidic environment is called an acidophile. These temperature and growth-based restrictions on microbes and where they can grow and in what conditions are mostly based on enzyme function. So enzymes are proteins in cells that help to drive chemical reactions that are essential for life. The shape of an enzyme, as shown in this figure as a red kind of Pac-Man looking figure, is very important. So if the 3D shape of the enzyme is optimum, it will fit the substrate, which will be turned into product, very tightly. If the temperature or pH at which the microbe is growing is altered, this will affect the shape of the enzyme. So we have on the right the red enzyme has changed shape slightly, so it doesn't bind to its substrate as well as it does at optimal temperature, and therefore reactions will not be occurring as quickly in the microbe, and therefore the microbe will grow more slowly. So the temperature and pH-based growth restrictions are mostly based on enzyme function, where enzymes function best at the optimal temperature and pH for the microbe. Another thing to consider when culturing or growing microbes in a laboratory is what osmotic pressure they require. So if we have a microbe in a low salt environment, like on the left, the sodium chloride percentage is 0.85, so very little salt outside of the cell. The microbe will look as we expect, but if we increase the salt content of the extracellular environment, so now we're at sodium chloride concentration of 10%, this will cause a lot of the water to leave the cell, and the cell will shrivel. We have the shell of the cell wall remaining in brown, but the plasma membrane and the cytoplasm and all the contents will shrivel up. So on the right, we have very high osmotic pressure, which will result in this shriveling known as plasmolysis, and the microbe will likely die. If there is a microbe that can tolerate very high salt concentration, so it would not undergo plasmolysis in a high salt or high osmotic pressure environment, this type of microbe is called a halophile. Another consideration for microbe growth is whether or not they require the presence of oxygen. If a microbe is aerobic, then it grows in the presence of atmospheric air and requires oxygen for growth and metabolism. If a microbe is anaerobic, it requires an area growing without atmospheric air and oxygen-free conditions to live. And this is based on metabolism for microbes, so how they extract energy from cells to make ATP. And we'll talk more about this, but just to introduce, there are 3 types of metabolism in cells. So we have aerobic respiration shown on the left, we have anaerobic respiration shown in the middle, and we have fermentation shown on the right. So for these 3 means of doing metabolism, aerobic respiration requires oxygen, where anaerobic respiration and fermentation do not require oxygen. So depending on the type of metabolism a microbe does, this will determine whether or not it requires oxygen for growth. So if we grow different groups of microorganisms in an agar-deep tube, which is the the test tube with a very deep layer of agar. At the surface of the agar, we have atmospheric oxygen level, but as we go deeper towards the bottom of the tube, the oxygen concentration comes to 0. So we're aerobic at the top of the agar-deep tube, and we're anaerobic at the bottom. So if we grow organisms in an agar-deep tube, we can categorize them into 5 groups based on their oxygen requirement. So in category A, we have obligate aerobes, which will grow only on the surface of the agar-deep tube. In group B, we have facultative anaerobes, which will grow most densely on the surface of the tube in the aerobic part of the agar, but will also grow in the bottom anaerobic parts of the agar. Category C are microbes that are obligate anaerobes, so these grow solely in the bottom anaerobic portion of the agar-deep tube. Category D are aerotolerant anaerobes, which grow evenly throughout the tube in the aerobic and anaerobic portions equally. And category E are microaerophiles. So these are microorganisms that require oxygen, but they require oxygen at a lower concentration than what is present in the atmosphere. So there are 2 things at play here. The first is what type of metabolism these organisms do. So any of the organisms growing on the top of the tube more densely will be doing aerobic respiration. So aerobic respiration is done by obligate aerobes, facultative anaerobes, and microaerophiles. All of the organisms that are growing in the bottom of the tube are doing anaerobic respiration, or fermentation. So facultative anaerobes are capable of these anaerobic metabolism forms. Obligate anaerobes are doing anaerobic metabolism, and the aerotolerant anaerobes are doing anaerobic metabolism. So of all of these, B, the category of facultative anaerobes, are capable of both aerobic respiration and anaerobic respiration or fermentation, whereas the other categories of microbes will do one or the other. There's another factor that we have to consider, and that is the presence of enzymes in the microbe cells that can detoxify oxygen. So in some forms, oxygen can be damaging to molecules in the cell. So cells have to have enzymes to alter these damaging forms of oxygen so they are not going to cause problems for the cell. So the second question, the first one being what type of respiration do this microbe do? The second question is, does the microbe have these enzymes for detoxifying oxygen? So in this figure, the groups that do have enzymes for detoxifying oxygen are obligate aerobes, facultative anaerobes, aerotolerant anaerobes, and the microaerophiles. And these enzymes are called superoxide dismutase, or SOD for short, catalase, and peroxidase. So any organism that can grow in the presence of oxygen has to have these enzymes for detoxifying oxygen. The dotted arrow for microaerophiles is meant to demonstrate that microaerophiles have these enzymes, but they are not extremely effective and efficient. Therefore, the microaerophile can handle oxygen, but only in small concentrations. With microbe growth, when we talk about a microbe growing, we're discussing the microbe growing in number, not size. So microbe growth would be an increase in the number of microbe cells and not in the size of these microbe cells. So microbes grow by binary fission, which is the way one microbe reproduces and divides into two daughter cells. So in this figure, we have a parent cell at the top, which copies its DNA chromosome. So we have two copies. Those two copies move to opposite ends of the cell, and the cell pinches off to form two separate daughter cells. So in this process, the parent cell and the two daughter cells are identical. The amount of time it takes for one cell to become two is called the generation time or the doubling time. So when one generation time has passed, the number of microbes in a culture will double. So if you look at microbe growth over 5 generations, if we start with one cell, we have, after one generation or one doubling time, that one cell splits into two. Then after a second doubling time, those two cells split into 4. After a next generation, we have 8, then 16, and then 32. So in a very short amount of time, bacterial growth will be very large. So we'll have large numbers of cells in a small amount of generations. So when we record bacteria growth data, we do so on a logarithmic scale. So shown again here is starting with one cell in the first generation on the left, and that cell divides by binary fission into 2. Then each of those cells in the next generation divide into 2, so we get 4, and so on. So if we look at number of cells over time, we have the number of cells increasing very quickly. So the best way to represent this increase in cell number is exponentially. So if we have every generation the cells will double, we can do 2 raised to whatever generation we're in. So for example, after 3 doublings, we're in the 3rd generation. So we take 2 for doubling every generation raised to the 3rd, because we're in the 3rd generation, which equals 8 cells. So after 3 generations or 3 doublings, we would have 8 cells. So using exponentials, we can calculate the number of bacteria after many, many generations. So representing this data on a graph is usually done like the graph in B, where on the x-axis we have time, and on the y-axis we have number of cells, and the number of cells is shown on a logarithmic scale. So where we are at 5, this is actually the 5th generation. So we are taking 2 raised to the exponential 5, which is equal to 32. And so at the 10 hash mark, we're at 2 raised to the exponential 10. And we've gone through 10 generations, 10 doubling times. So we can represent very large numbers that start being created after only a few generations on a graph, but still see the small numbers of early generations. If we try to draw bacterial growth data on a linear scale, as in part C, where each hash mark is the number of cells, so 200,000 increments, you can see that after 20 generations, we're up to about 1 million cells, just like we were in part B, but it's very difficult to see what's happening in earlier generations. So at any point less than 15 minutes on this scale, it looks like nothing is happening, although we are certainly getting increases in bacterial growth. So I have on these next 2 slides 2 sets of data. So we have a linear plot first of the data where we have that telltale curve that shoots up very quickly. And then on the next slide, we have the same data plotted on a logarithmic scale. So you can look at both of these on your own. The doubling time for these bacteria is 20 minutes, and we're going through 12 generations in both cases. So the point being that plotting bacterial growth data on a logarithmic scale is much better than the linear scale. Bacterial growth in a culture in the lab goes through 4 distinct phases. So we have over time on the x-axis and log number of cells on the y. If we put bacteria cells in a tube with media which has all required nutrients, initially we would have a phase where there's no change in cell numbers called the lag phase. And at this phase, the bacteria are preparing to divide. So they're replicating DNA, they're increasing in volume, they're replicating cellular organelles and increasing volume of cytoplasm. So although numbers don't change in the lag phase, the bacteria are very metabolically active. The next phase is the exponential growth phase, and because the y-axis is a logarithmic scale, the straight line tells us that the growth during the exponential phase is constant. So during the exponential growth phase, we get the largest change in cell numbers. The third phase is the stationary phase. At this point in the culture, we start to get a number of dead cells, which is represented in the gray. So a fraction of the culture is now dead because nutrients are being depleted and waste is building up. So the number of cells that are dividing is equal to the number of cells that are dying in the stationary phase. So overall, we have no change in cell number. And then the fourth phase is the death phase, which represents a decrease in cell number. So the number of cells dying is greater than the number of cells replicating. And so we have the straight line going down. And since we're on a logarithmic scale, the straight line tells us that the death rate is constant in this culture. So if you put bacteria in a culture, they will go through each of these 4 phases. The timing for each phase will depend on the bacteria culture that you're growing. There are several ways to count bacteria cell numbers. So one of them is the batch culture method, where you incubate media with bacteria cells. You allow the media to grow over a period of time and at different intervals. So in this figure, at every 60 minutes, we take a sample of the larger culture in the flask. We put that sample on a petri dish and we allow the petri dish to grow for 24 hours until we see the appearance of bacterial colonies on the surface of the agar. You would then count the colonies, and that would give you a representative number of how many bacteria are present in the original culture flask. The benefits of the batch culture method are you count only live cells. One of the disadvantages is that it takes time because you have to allow the petri dishes to grow overnight, so results are not instantaneous. We also have a way to count bacterial cell numbers by a lab machine called the turbidometer, which measures how cloudy a liquid culture is. So if a liquid culture is very clear, there's very little cell growth inside it. And in the turbidometer, a light is shined through the liquid culture, and the percent of light transmitted through the culture is measured. So when all of the light is transmitted through the culture, it means there's low cell growth, and the turbidometer would report low growth. If the liquid culture is very cloudy, then there's a lot of cell growth. So if the turbidometer shines a light through the very cloudy liquid broth, then the percent transmitted light would be very low because a lot of the light would be deflected by the cloudy media, and so the turbidometer would report high growth. The benefit of the turbidometer in counting bacteria cell numbers is that it's very quick. One of the downsides is that it cannot distinguish between live or dead cells. So in a highly turbid culture or very cloudy culture, there could be presence of live and dead cells both. Another way is microscopic cell counts. So this is done with a special slide that has a grid etched into the surface. So you put a drop of your bacteria culture onto the grid, and you would count cells that are present on the grid, which just helps you to stay oriented in the grid and make sure you don't count the same cells twice. So after adding the drop, you would look under the microscope and you would see the magnification shown in the circle, and you would use that to count through cells. The advantage of the microscope count is that it's very quick and very easy to do. A disadvantage is that it can be hard to distinguish live from dead cells. It is also very difficult to count cells that are motile and that are capable of moving around because they'll move on the grid and will be hard to keep track of. So that concludes this set of lecture slides. You are now ready to do your homework 2.